Cambridge AS Biology. Chapter 1, Cell Structure. Biosandwish. 9700 Bio in 5 minutes. Variant 23. Year 2018.
Aphids are small insects which feed directly on phloem sap. The salivary glands of aphids have secretory cells that make and release a variety of proteins that assist in feeding. Figure 1.1 is a transmission electron micrograph of a small area of a salivary gland cell of an aphid. Describe the role of Golgi bodies in secretory cells, such as the salivary gland cells of aphids.
Aphid is an insect with piercing sucking mouth parts that feed on organic matter like sucrose and amino acids in phloem sap.
The Golgi body collects, processes and sorts molecules, particularly proteins from the rough ready for transport in Golgi vesicles either to other parts of the cell or out of the cell, secretion. Glycosylation. Golgi bodies add sugar molecules to either protein or lipids to form glycoproteins and glycolipids, which are needed as receptors at cell surface membrane. Golgi bodies are responsible for cutting polypeptide chains like C-peptide section from proinsulin hormone and removal of the first amino acid, methionine, from newly formed proteins to make a functioning protein, active insulin hormone. In plants, enzymes in the Golgi body convert sugars into cell wall components in the form of vesicle that fuse together at equator of the cell during cell division to form middle lamellae between the two daughter cells. Explain why secretory cells have large numbers of mitochondria. Mitochondria release ATP molecules. Secretory cells need a lot of ATP required for 1. Protein synthesis starting from mRNA transcription. 2. Translation of mRNA in polypeptide chain by ribosomes. 3. Besides movement of vesicles containing proteins on microtubules of cytoskeleton. 4. Finally, exocytosis of products. 2. Mitochondria are partly controlled by the nucleus, but can also function independently. Suggest the features of mitochondria that allow them to function independently of the nucleus. Mitochondria have its own, circular, DNA-carrying genes. mRNA transcribed from mtDNA. Mitochondria produce their own tRNA. Can replicate DNA. These genes, when expressed, they form its own proteins, as respiratory enzymes and ATP synthetase. Mitochondria have its 70S ribosomes for translation. Mitochondria can divide by binary fission, not by mitosis.
Mitochondrial DNA is circular, not complexed with proteins. Carry a few genes. Many copies inside each mitochondrion. While nuclear DNA is linear, complexed with proteins. Carry all genes, that is called genome.
Aphids are important vectors of plant viral diseases. Describe the structure of a typical virus. Viruses are acellular, they are very small particles, range from 15 nanometers up to 1000 nanometers. The virus particle contains a genetic material, either DNA or RNA. Lie in the core and surrounded by a protein coat called capsid. The capsid is formed of many small units called capsomeres. It may be surrounded by phospholipid bilayer gained from its host cell during release from it. 2. Suggest how viruses are able to pass from one plant cell to the next without crossing membranes. Viruses pass through plasmodesmata. Plasmodesmata are channels in the cell walls that link adjacent plant cells together. It allows transport of substances and communication between cells. Viruses may pass via cytoplasmic strands via symplast pathway. Thank you for watching.